Well, thank you very much, Leonard. And Orla, we don't have time for questions. Perhaps after the last speaker, Dr. Chen, we change our topic from ALS to neuropathic pain. Dr. Chen is from Stanford University, California, USA. Her research is focused on the impact of homeostatic plasticity on Hebbian plasticity and how this interaction influences in animals. And her talk is about retinoic acid receptor signaling in neuropathic pain. Thank you very much.
Thank you. I'd like to thank the organizers for inviting me to this meeting. And a disclaimer, I don't work on ARS, and my closest encounter with ARS was when Tom, after the Nobel Prize, decided to enter the ice bucket challenge. I was helping with carrying the ice and dumping it over his head. Instead, my lab works on, majority of my lab works on the molecular mechanisms and interactions between different types of plasticity, synaptic plasticity, and how they contribute to our cognitive function. But today, I'm going to touch base on a very more fundamental perception or a more fundamental function of our CNS, the pain perception. And this, actually, the talk today is my first adventure into the spinal cord, the part of the CNS that I've never worked on. But the signaling mechanism has everything to do with what we have been working on in the brain. So let me see. Yeah, so we all know acute pain. When you cut your finger, scrape your knee, or break a bone, you feel the pain. And it's very important that we have this perception because it tells our brain something's wrong. We have to do something about it, otherwise we're in trouble. It's an evolutionarily protective mechanism, important for survival. However, this perception sometimes can develop into a pathological condition that counteracts the survival instinct and therefore becomes harmful. So neuropathic pain is a form of chronic pain that persists long after the recovery of the initial injury has already recovered. And it's usually induced by peripheral nerve injury. Some of the characteristics of neuropathic pain is that it's spontaneous. So without anything touching you or hurting you, you could feel the pain. Or it has a phenomenon called allodynia, which is a normally gentle, innocuous stimulation. For example, a gentle touch, which could be even pleasant, could induce excruciating pain in patients. Or you have increased pain sensitivity, also called hyperalgesia, for something like a pinch, which is unpleasant but not that bad in patients with neuropathic pain, could induce massive amount of pain perception. A common thing of these different forms of pain is that they are usually resistant to common painkillers, and it's very difficult to treat them. And one of the obstacles in treating neuropathic pain is because we don't really fully understand the neurobasic mechanisms underlying the development or the quantification of this phenomenon. So a very quick introduction to those of you who are not familiar with the pain pathway or pain perception. So we have nociceptors that the cell body lies in the dorsal root ganglion, and they have one side of the nerve ending goes to the tissue, innervates the underneath of skin or inside organ. So this is help you to perceive where the pain starts. The other end of these neurons goes into the spinal cord, projects to the dorsal horn, and make the first connections to the CNS to send the signal. And then in the spinal cord, the signal gets relayed and across the midline to the contralateral side and goes upwards, thus forming the ascending nociceptive pathway. So along the way, it makes connections to the brainstem nucleus, to the midbrain nucleus, and eventually to the cortical regions, for example, the somatosensory cortices, the anterior cingulated cortices, and also to other regions of the cortices and the subcortical regions that are important for affective aspect of pain, because pain not only causes the perception of pain, but it causes people to feel depressed, anxious, right? So there are many aspects of pain perception that I won't be touching. In order to study neuropathic pain, animal models has been developed to mimic this kind of chronic condition. They all involve one form or another of lesion or peripheral nerve injury. So I'm listing a few of them here. So, for example, spinal nerve ligation is to basically ligate the entire bundle of spinal nerve that innervates the lumbar 3 or lumbar 4 to 6, or you can dissect these branches. For example, the ones that I'm using in my research is called a spare nerve injury, which is we take apart the sciatic nerve, which has 3 branches, and we ligate and cut 2 of them, and we only save one branch, the serial nerve, intact. The serial nerve would innervate all these lumbar 3 to 5 regions together with these 2 nerves that are already cut. So you create a model where there's partial nerve injury, but part of the input is still intact. And these are the conditions that it generates neuropathic pain. So this model was first developed by the Wolf Lab more than 20 years ago, and this is what we did in our surgery. We isolate the sciatic nerve and find the 3 branches and tie up a knot on the 2 of them and make a cut distal to the ligation site. And then this is the spare nerve, and that's why it's called the spare nerve injury model. And the way that we assay chronic pain, or we call it mechanical allodynia, is to use Van Fry filament, which is basically a series of elastic filaments that there are with different diameter, made of slightly different materials, that when you touch the bottom paw, the bottom of the animal hind paw, and touch it so much that the filament eventually buckles elastically, it exerts a fixed amount of force to the paw. And so if the animal feels this force is too painful, they'll withdraw their paw, right? So you detect the threshold of the force that's being put onto the paw and ask, what is that withdraw threshold? Now, in normal animals, they will eventually withdraw. Like if I poke you with a pencil, and if I poke it hard enough, you will feel the pain and you will withdraw your hand. It's the same thing. But for animals with mechanical allodynia, they will withdraw at a much lower force. So this is an indication of hypersensitivity to the mechanosensory input, and therefore a way of assaying mechanical allodynia. So under normal perception, the pain pathway and the mechanosensory pathway have their own sort of circuits. So they each activate different input neurons. So the painful stimulation, like harmful chemicals or high or low temperatures or sharp pointy objects, would activate nociceptors, and they will directly project to the pain-projecting neurons in the dorsal horn of spinal cord. And this gives you the pain sensation. However, harmless touch would only activate low-threshold mechanosensory receptors, and they will activate the touch-projecting neurons. These 2 pathways normally don't overlap with each other, but there is a branch you can see that's here. This is to ensure that when you have extremely high activation of mechanosensory input, when someone pinches you really, really hard, you're going to feel the pain. It's because this mechanosensory input now this branch goes upwards. Not only it activates some of the subcutaneous nociceptor terminals, it also ensures this pathway gets opened. But normally, this pathway is gated by this blue neuron here, is an inhibitory interneuron that makes sure this flow of information is closed. That's why it's an inhibitory gate. So what happens under neuropathic pain condition is that there's a central sensitization. That is, this pathway, the pain pathway itself, gets hyperactive. So with the same kind of painful stimulation, you feel more pain. But additionally, this flow of information that should be blocked is no longer blocked because the gate is now open. And this is because the inhibitory interneuron function is somewhat compromised. And that's been shown that if you just inject GABA into the spinal cord, you could actually temporarily block the neuropathic pain. Or if in a normal animal, if you inject GABA receptor antagonist, you could induce temporary pain. Okay, so this means the gate itself is very important for controlling this information, and this misdirection of the mechanosensory information flow to the pain perception, to the pain pathway, is one of the main cause of neuropathic pain. So my lab, as I said, works on homeostatic plasticity and among many other forms of plasticity. So what is homeostatic synaptic plasticity? It is a form of plasticity that ensures neural network stability. So when you have neurons under perturbation condition, either through synaptic strengthening or through excitability changes, it could become more excitable. And when the whole network becomes more excitable, it becomes unstable. So homeostatic mechanisms, by involving either changing intrinsic excitability or modifying synaptic excitation or synaptic inhibition, can restore neuronal activity to the optimal range and therefore regain stability of the network. But my lab also believes that the function of homeostatic plasticity is beyond just restoring network stability. It actually ensures that there's a coordinated modification of synaptic excitation, inhibition, and even excitability, so that not only the neural network stability is maintained, its ability to preserve the capacity for optimal information processing to best represent external world must be preserved. So we've been working on the mechanisms of homeostatic plasticity for the last 15 or so years, and so this is by no means a complete picture of the pathway we worked out, but the major question, message here is that we've discovered a master player, the receptor-alpha, that plays an important role post-developmentally. This is a molecule that mediates transcription regulation during development. It's considered a developmental morphogen. However, in mature neurons, it goes out of the nucleus, goes into neural dendrites, and maybe even presynaptic axon terminals, and to actually regulate protein translation, and participates very actively in homeostatic synaptic plasticity. So, for example, it regulates AMPA receptor synthesis, and can compensate for the lack of activity by inserting more AMPA receptors postsynaptically. We recently also just submitted a study to show that it regulates retrograde messenger BDNF synthesis postsynaptically, which then acts on the presynaptic TrkB receptors to enhance synaptic transmission as another way of compensating for reduced neuron activity. So we then hypothesize that in the case of neuropathic pain induced by peripheral nerve injury, perhaps it's the excessive homeostatic synaptic plasticity resulting from the loss of afferent input that triggers the overcompensation and hypersensitivity in the CNS, in the dorsal horn circuit. And these compensation can initially occur in the spinal cord circuit, but then later on triggers additional downstream compensations in the central ascending pathways, for example, in the cortical regions. So this is the core circuit. It's by no means incomplete because spinal cord dorsal horn circuit is actually really, really complex with many, many different types of neurons. But this is the main core circuit. And so the touch information goes, activates the low threshold mechanosensory receptors, which is also called A-beta fibers. They comes in here, and their goal, they send projections to somewhere which is not in this diagram, that goes to the sensory, the touch mechanosensory projection neurons that gives you sensation of touch. But they also send the branches up, and through these excitatory relay neurons to connect with the nociceptive projection neurons. This will give you the pain sensation. However, this flow of information is usually gated or shut down by the inhibitory local control of the PV positive GABAergic neurons. So feed-forward inhibition by PV neurons ensures no erroneous flow of information from normal touch to the pain perception, to the pain pathway. This is very much similar to what we find in a lot of the cortical circuits. For example, in the visual cortex, if you look at how the information flows, the peripheral vision-activated information goes into the cortical region through activating principal neurons in layer 4, and then it will send information to layer 2 principal neurons. But there's also a feed-forward inhibition here, mediated by PV neurons. And this feed-forward inhibition is very important for controlling the formation of receptive fields. So our lab and others discovered that in the visual cortex, if you give visual deprivation, in this case binocular enucleation for just 3 days, you will induce robust homeostatic compensation in the visual cortex in the layer 2-3 neurons. But very surprisingly, there's not much change in the excitatory synapse transmission, but there's a profound reduction in the inhibition. And furthermore, we showed that if we delete IA-alpha, the master molecule master mediator for homeostatic plasticity from PV neurons specifically, we were able to completely block this homeostatic downregulation of inhibition. And this is not the case if we remove IA-alpha from CCK positive inhibitory interneurons. So it's neuronal type specific regulation. So to recap, what we found in our previous cortical study is that input or sensory afferent deprivation induced homeostatic plasticity would weaken PV output as a form of homeostatic compensation. And this requires IR-alpha expression in the PV neurons. So coming back to the neuropathic pain question that I would just alluded you to earlier, can this same mechanism operate in the spinal cord dorsal horn circuit? So we took PV-CRE driver line, and we verified its expression, specificity, and efficacy in both ACC, S1, and dorsal horn neurons, and they look significantly high enough. We then crossed it with IR-alpha flox mice, so we could specifically delete IR-alpha molecules from PV-positive neurons. And so this is using RNA-scope to look at deletion specificity, and you can see that with RNA-scope, we can see that in conditional knockout dorsal horn neurons, there's a significant reduction in the IR-alpha expression in PV-positive neurons. We also performed a single-cell QRT-PCR by patching on these PV-positive neurons, and we saw a significant reduction or deletion of IR-alpha. So here's a key experiment. Can these animals develop neuropathic pain? So if you look at the green symbols here, this lighter green symbol is the control side. That is the unlesioned side. After the lesion, there is no change in the withdrawal threshold for mechanosensory stimulation, indicating there's no pain, excessive pain perception. But the lesion side, the hind limb withdraws much earlier. It's showing you the sign of mechanical allodynia. However, if we delete IR-alpha globally throughout the whole CNS from PV-positive neurons, the mechanical allodynia is prevented. And this is not the case if we do the control experiment by deleting IR-alpha from SST-positive inhibitory interneurons. So there is something unique about PV neurons. And so what about other type of neuropathic pain? So we looked at temperature, thermal sensitivity. We looked at long-term versus short-term inflammation-induced nociception. None of them were affected. So this pathway, this PV-mediated inhibitory gate, is very uniquely involved in the mechanosensory allodynia. So we then looked at more closely all the ascending pathways from the spinal cord upwards. So we use phos staining to look at neuron activation. So on top is the wild-type neurons. This is the injured side. You can see without any stimulation, there's already hyperactivation of neurons. This is the dorsal horn part. This is sort of the middle part of the spinal cord. This is the contralateral side, nothing. Ipsilateral side, there's a lot of signal. In the IR-alpha knockout mice, the signal is eliminated. Now, if you go on to the brainstem lateral parabrachial nucleus, remember this is where the signal crosses the midline. So in the wild-type animals, you see the contralateral side. There's elevated activation of ascending pathway, but in the knockouts, this is gone. When we keep going up to a few cortical regions, in the cortical regions, there's actually bidirectional projections. So you can see that contralateral, but also to less extent, the ipsilateral S1 is activated. This is the hind limb region of the mouse. And both signals were reduced in the deletion mice, ACC as well. So if we quantify multiple brain regions, you can see general pattern. That is, in the SNI injured animals, the ascending nociceptor pathway activation is heightened. And this hyperactivity is blocked by deletion of IR-alpha from all PV-positive neurons. And it doesn't affect the formally induced inflammation-induced pain. So this is, again, very specific for mechanosensory. We then looked at layer-specific distribution because you can see this activation is throughout the dorsal horn, not just in the superficial laminar where the nociceptive projection neurons are. So you can see the activation, the hyperactivation is happening everywhere. And in every single laminar, you can see reduction, significant reduction of the activation by IR-alpha deletion. We then further confirmed that by injecting retrograde dye fluoro gold in the LPBN, which then is the first relay from the spinal cord nociceptor projection neurons, so we can retrogradely label projection neurons only in this case, which is shown yellow here, and we quantified in the wild type of knockouts, and you can see the knockouts also specifically reduced the activation of nociceptor projection neurons. So this explains why there's less pain perception, why we could block mechanosensory mechanoallodynia. So, but what is the cellular mechanism for the IR-alpha function in nociceptive neuropathic pain development. So in the spinal cord dorsal horn, majority of the PV neurons are expressed in lamina 3. So it's basically here. And some of it is, so here what I'm showing you is the PV neuron that are PV crossed with a reporter line, so that all the YFP is indicating where the PV neurons are. So this is no longer dorsal horn. Dorsal horn ends about here, okay? So you can see that most of the neurons are in lamina 3, some of them are in inner lamina 2. So when the earlier histology studies from David Hughes' lab showed that the afferent, which is primary afferent, which is labeled by the V-GLUT1, labeling these red puncta here, overlaps with both PV neurons and the PKC gamma, which is a form of excitatory interneurons that relays these information flow upward to the nociceptor receptors. So these are the contacts these afferents is making, both inhibitory neuron and excitatory interneuron. But if you look at where the PV terminals are, it also contacts directly the excitatory relay neurons, which is this kind of synapse. It also contacts the afferent input, which forms a presynaptic sort of partate synaptic structure. I'll give this a little bit more attention later, so don't worry if you don't see. So here it is. The PV neuron would synapse onto the presynaptic terminal of the glutamate terminals of the afferent fibers. And these are the ones that release glutamate that activates postsynaptic neurons and sends the information flow forward. This presynaptic inhibition shunts the voltage or the current in the glutamatergic terminals and prevents the glutamate from releasing. Therefore, in general, it is inhibitory. However, with physiological tricks, when you bring the temperature of recording, so at the body temperature, this is inhibitory action. The GABA release inhibits glutamate release and therefore blocks the afferent flow of information. However, if you lower the temperature, there is accumulating of glutamate because of the inefficiency of glutamate transporter, could actually help depolarize the terminal further and therefore induces glutamate release, which is very counterintuitive, but it becomes a very scientifically very helpful tool. It's a phenomenon called a primary afferent depolarization, discovered more than 50 years ago in the spinal cord circuit by some very early electrophysiologists. But it's become very useful in our hands that this allows us to assay the GABA release here by recording in this neuron here the glutamate release, because glutamate release is triggered by GABA release. So the amount of glutamate release or the amount of EPSC we record in this neuron here would tell us the amount of GABA that's released presynaptically from the PV neurons. So this is our experimental setup. Let me just check my time. Okay. So this is a PV neuron. We express channel rhodopsin to activate them optogenetically. And we can record both OIPSC here and OEPSC here. But I'm going to skip the details here. But by playing physiology tricks, we could isolate EPSC and IPSC separately. So we could look at these two forms of inhibition separately. So because this is very heterogeneous population of neurons, and postsynaptically, we're patching blindly, only in the region of lamina 3. So for each neuron we patch, we suck the content out. We do RT-PCR to verify a few genetic markers. This gives us some way of categorizing neuronal types, and this is what we have. 40% of them are not responsive, so they are excluded from analysis. 27% are PKC gamma positive, which has been reported. But we've also found nearly 20% of the neurons are V-GLUT3 positive. They are also targets of PV neurons, which hasn't been reported. And there are some other smaller populations which we also excluded because their N number is too small for analysis. As you can see, this is a two-step synaptic release. So the synaptic delay is actually longer. And this is shown here, that there's a 12 millisecond on average synaptic delay from the light onset to the onset of the response. And the response examples are shown here. The reason we separate these by using genetic marker, qPCR genetic marker assay, is because the response size are very different depending on the target. So the PKC gamma positive neurons has a much smaller synaptic response, or EPSC response, compared to the V-GLUT3 ones. If you put them together, you'll never see any phenomenon, right? However, in each different type of neurons, you can see a clear reduction in the GABA release assayed by reduction in the OEPSC response in the injured side. And in both cases, the IR for deletion can completely block this reduction. So in this form of presynaptic inhibition, I-alpha deletion itself prevents the reduction of inhibition. And this is the second form of inhibition, directly onto postsynaptic side, which is very traditional. GABA release activates GABA receptors postsynaptically. And we did the same kind of experiments, the same qPCR for every single neuron we patched, and the same conclusion that the PKC gamma positive neurons and the T3 positive neurons, despite their difference in the response amplitude, show nearly half of the reduction, half of the iPSC response after nerve injury, and this was blocked by alpha deletion. And notice here, the synaptic delay is only 4 milliseconds. This is a monosynaptic response. Anyways, so this is all very nice, but the experiments were done in the global deletion of PV RA-alpha. So how do we know that this process starts in the spinal cord? How do we know the phenomenon we recorded with physiology is relevant to the behavioral output we see here? I'll have just a couple of slides left. So here we use a genetic intersection method where we ordered PV flippase mice crossed with I-alpha flox mice. And then in the spinal cord, the lumbar 3 to 5 region, we injected AAV that express a flippase-dependent Cree. So this allowed us to delete I-alpha specifically from PV neurons in the spinal cord. And these mice did not develop neuropathic pain after SNI. And not only that, but also this completely reversed the hyperactivation of all the regions above spinal cord in the ascending nociceptor pathway, suggesting this is the true culprit of the initial quantification site for acute pain transition into chronic pain. And if we do the same experiments in ACC, it doesn't change anything, so there's no effect. If we do the same experiments in S1, we also didn't see any effects. Sorry, I went through it too fast. So this is indeed one evidence showing that the quantification site initiates in the spinal cord. We then went on a different approach. We used a osmotic pump to deliver a retinoic acid receptor antagonist, the Rho 41, 52, 53, locally to the spinal cord at the time of injury. We start the pump. And this pump went on for 4 weeks. So this is a control vehicle just showing the pump itself doesn't impair the development of mechanical allodynia. However, with the pump infusing RA-alpha antagonists, we prevented the development of neuropathic pain. But not only that, when the pump runs empty, when there's no more blockade, RA-alpha function recovers to normal, the pain never comes back. So we were able to, by briefly blocking RA-alpha function, to briefly basically prevent the switch of the gate open, we could prevent neuropathic pain development permanently. So that's my summary slides, that during the normal condition, health condition, a gentle touch would send information through the low threshold mechanosensory receptors that activates PV neurons, which then immediately shuts the gate so the information doesn't go up, and you don't feel the pain, you're happy. But under the peripheral nerve injury, this inhibition is weakened, as you can see, and therefore PV neuron is no longer able to block the flow of information that goes this way, and therefore the nociceptor pathway is activated, and you feel the pain. So our future direction involves multiple, there are so many questions. This is just our beginning effort, literally the first 3 years of us working on spinal cord. And we want to know how does presynaptically I-alpha functions to regulate synaptic release. Is there a critical period? We infuse the drug for 4 weeks to prevent the development of neuropathic pain. Can we shorten it? Is there a critical period that with a minimum blockade of I-alpha function, we could permanently prevent the development of neuropathic pain? And does the same homeostatic mechanism in superficial laminar inhibitory interneurons drive thermal hyperalgesia? Can we find a different set of circuit but use the same mechanism to control the thermal hyperalgesia? And what mechanism may underlie cortical plasticity? So after the spinal cord become the first step of quantification of the pain, what happens in the ascending pathway? What happens in the cortex that further solidifies the quantification of neuropathic pain? So this is my lab. This is a picture taken. I love to show this picture. This is what we did in the pandemic. We did all the lab meetings on Zoom. Everyone was working in the lab with masks on, but we kept the research going. This study was mostly conducted during the pandemic, and this is the hero behind the study, Bing Cao. And these are the funding agencies. And I like to give special thanks to my children, it was not easy to work with young children and as a full-time professor, but I really enjoyed having them, and I really thank them for their enthusiasm for my science. They are the biggest fan to my science, so thank you for being there for me.
Well, thank you very much. Well, if we will be on time. We don't have time for questions. So thank you for the three speakers, two different topics, neuropathic pain. Very interesting for me because I am working with patients with traumatic nerve injury, neuropathic pain, and chronic pain.
I see.
Thank you. And thank you very much to Orlan Leonard, focus on ALS. We have 15 minutes, just 15 minutes, and come back again. I hope to see you, no? 10 minutes? 10 minutes, just a coffee, one question, and come back again. Thank you very much.